In case of PCR, we know that we are performing PCR amplifications, and this amplification is desired for a particular gene of our interest. It may not always be a gene, but it may always, some other time, it may be some promoter, some other regulatory sequence, or some terminator, or there may be several different desires, but the technology is the same. And we need a template for that purpose. And the template for a PCR reaction can be a double-stranded DNA, it can be a single-stranded DNA, okay? Double-stranded DNA is usually the genomic DNA isolated from an organism. It may be mitochondrial, it can be chloroplast, or it may be a total genomic, which includes all the three. Remember, the word total genomic means all the types of DNA which are present in that organism, whether mitochondrial, chloroplast, or genomic. Then when we say genomic DNA, it means it's only from the nuclei, okay? And then when we say single-stranded DNA, it is either the cDNA, which we prepared from messenger RNA by using reverse transcriptase enzyme, okay? Or it can be from any virus, which is single-stranded DNA virus. We usually don't use RNA as a template because RNA gets degraded very quickly. Okay? It's broken down into small pieces. But I have seen in my experience that if we take directly the RNA as a template, we can still get some amplification. Okay? If the quantity of the total RNA is good, the RNA is not degraded initially, so you can try actually to PCR amplify from RNA, okay? It works, but it does not work as good as a DNA molecule. Okay? Now, if you look at the template, usually in the pictures, they represent double-stranded DNA, but consider that this may be double-stranded or this may be already a single-stranded. If you're starting with a double-stranded DNA, you need an additional step, and that is the denaturation step. Do you need a denaturation step in case of single-stranded DNA? The answer is yes. Because whenever it is single-stranded DNA or single-stranded RNA, it gets into a secondary structure formation. And you need to open that secondary structure by heat denaturation. So heat denaturation is always required, whether it is a single-stranded DNA or it is a double-stranded. Because we have gone through this lecture already, and I have mentioned that it is just a procedure, but understanding what is going on in this procedure is very important. So I will concentrate mainly what is going on, besides the procedure that I will discuss briefly with you. Okay? Any DNA replication mechanism, it requires a DNA polymerase. In this case, we have a DNA polymerase which is very susceptible, or which is very resistant to high temperatures, and that's tag DNA polymerase. Now the question is tag DNA polymerase the only polymerase which is used in PCR reaction? The answer is no. Tag is just one enzyme that can be used in a PCR reaction. Remember, this is not a high fidelity enzyme. It makes mistakes more frequently. But it tells you, if you're working with diagnostics and you want to see whether the infection is there or not, tag is okay. You can work with tag. If you are working with cloning a gene or a regulatory sequence, you don't want to introduce any mutations in the gene sequence. So tag is no. not the enzyme of choice. Then there are some alternate enzymes, and you already read about those enzymes, DNA modifying enzyme, that is PFU DNA polymerase, which is a high fidelity DNA polymerase. It makes much less number of mutations in the polymerization reaction as compared to the Taq polymerase DNA. Okay, so these are some of the basics that you must consider, that when you are trying to set up a PCR reaction, think about your template and then what you are going to amplify and what kind of the polymerase is the best one for that. So select the one which is high fidelity if you're working with the genes. If you're just working with the detection mechanisms or optimization of a PCR reaction, you can go for Taq polymerase. And then we come to the procedure. Template is one of our requirement. Taq polymerase is the second requirement. Then we need primers, because primers, they are always required for any sort of DNA replication mechanism. Okay, this enzyme, whether it is Taq polymerase or PFU polymerase, it always requires a divalent cation. In case of Taq, it is magnesium chloride. Okay, the magnesium is the cofactor for this enzyme. So this is what you require. And then what else? The nucleotides will be required for incorporation by the DNA polymerase. If there is no nucleotide, the system will not work. The DNA polymerization will not take place. Okay? So these are some of the basic requirements which every student requires or everyone requires before setting up a PCR reaction. We add just water in there to get the optimum concentration of every constituent that we add into a PCR reaction. One thing that I routinely mention to my students is that whenever you are thinking of setting up a PCR reaction, you must assure yourself that you are not going to be disturbed during this reaction. All the other research methods also require your complete attention. Okay? But here we are actually mixing 8 different ingredients into a tube. So what you do in the beginning, I will go to that step later on when I will go to the individual ingredients of a PCR reaction. But you must understand the basic exponential amplification reaction. Okay? In a conventional PCR reaction, we usually don't consider this increase in the amplification through every cycle, but exponential amplification must be in your mind. That in the beginning, when you start, you have a few number of template molecules. And those templates, they act as the synthesizing machineries for the new strands of DNA upon them. So in this picture, what I have tried to show you is that initially, if we have 2 copies of the DNA, after one cycle of PCR is complete, okay, it's a polymerase chain reaction. It is a cycling reaction. So you're performing the same DNA polymerization again and again and again. Okay, so in the first attempt of DNA replication, you get 2 additional fragments synthesized. So the first cycle, after first cycle, what you get is 4 template molecules. The starter was 2 molecules. And that is called doubling from 2 to 4. And if you go for a second PCR cycle, these 4 DNA fragments should give you 8 fragments. Okay, so you can see again, it is a doubling reaction. So what theoretically it means is that every cycle doubles the amount of the template. This is the take-home message. That every cycle doubles the amount of template, and The efficiency of PCR reaction, we say, is doubling. Doubling means one template molecule is amplified to 2 with every cycle. And the efficiency of the reaction is therefore 2. That with every reaction, it is doubling. Okay? And after 10 cycles, the number of template molecules should be 2 to raise power 10. And after 30 cycles, it should be 2 to raise power 30. That is the formula. That is 2 to raise power n. N is the number of cycles. Okay? So, for example, if you have run your PCR for 36 cycles, starting from 2 template molecules, at the end of 30 cycles, you should have 68 billion copies. And that is PCR amplification. That you start with just a few number of template molecules and you end up with billions and billions of amplified products. So what it helps you? It helps you. You have so many copies of the gene that you can easily clone it. You can see it. Okay? So after 40 cycles, obviously it will be 2 to raise power 40. And I'm telling you this because we run the PCR reaction for about 35 to 40 cycles. It depends on the kind of template. If we have enough template in the beginning, it means 1,000 copies in the beginning. We can end up with 35 cycles. There's no reason to go beyond that. Okay? But as a standard, you can think that it's the 40 cycles per PCR reaction, okay, which is normally done in the laboratory. And I hope that you understand that what is 2 to raise power n. 2 is the doubling with every cycle, and n represents number of cycles. 2 here, 3 here, 4 here, 5 here, and so on. Okay? Now, if we now come down, that what is happening in a PCR reaction is that we have a double-stranded DNA, which is usually linear in form. So we give the first cycle at high temperature to separate the two strands of DNA or remove any secondary structure which may be associated with a single-stranded DNA, if you're using single-stranded DNA as a template. And this denaturation is the first step in a PCR reaction. The total steps are 3 in one cycle. First is denaturation at 94 degrees centigrade. Our time for 1 minute is enough during the cycles, but a preliminary time of 5 minutes when you start the PCR, just give a 5-minute denaturation. It will actually denature, high temperature will denature many of the proteins, small proteins which are associated with the DNA, and would help to the two strands to separate from each other at prolonged exposure to high temperature. Okay? In the remaining cycles, you can take this time to 1 minute. That is sufficient. Okay? Because most of the small proteins which are associated with DNA have already been degraded in the first 5-minute denaturation temperature exposure. Okay? It's not a cycle. It is just a single treatment at 94 degrees centigrade for 5 minutes, and then you can start your cycles on that. So what happens? The two strands, they get separated. They have the primers in there, and the primers only bind to the single strand if you drop down the temperature. The temperature drop to what temperature is dependent on the kind of primer that you are using. What is the GC content? What is the length of the primer? What is the melting point of the primer? So you are concerned mostly with the melting point, okay? And you must also know what is the GC content, because it is the GC content and melting point which tells you that what should be your optimum annealing point. I will talk about that later, okay? That how you calculate the annealing point for a PCR reaction. But hope that you are familiar now with the first step, which is the denaturation, that is separation of the two double-stranded DNA or removal of the secondary structure within a DNA molecule. The second point is annealing. Annealing is the binding of the primer with a single-stranded template DNA molecule. Okay, that happens at a certain temperature, which is called annealing point. Okay, how to calculate that? We will see that later. Okay, and finally, when the primer has bound to the template, it is ready for a polymerization reaction. Means that the polymerase cannot start synthesis of the second strand from 5' to 3' direction to make a double-stranded DNA in one cycle. And this is going to be repeated in the next cycles. That in every cycle, your DNA will be denatured, it will be annealed again by the primers, and then it will be polymerized again. So this is all. In a PCR reaction, technically, if you see into that, it's not more than that. Okay. The things which are important is setting up of a PCR reaction. And I said, just assure yourself that when you are setting up any molecular biology reaction, take out your notebook, write down the components of the reaction starting from 1, 2, 3, 4, 5, 6, as many as they are. Write those components just like this written here, okay? Target, 2 primers. Forward, you can write forward separately and the reverse separately. So the total number should increase to 8. If you are writing your forward primer separately and reverse, and make that habit. Keep them separated. Don't write them as 2 primers, okay? At position 2, write forward primer, at position 3, write reverse primer. And the total number that will be, you get 8. So after you have written down all these, make a habit that whenever you add, for example, target DNA in a test tube, in a PCR tube, okay? So make a tick over there. Even if you get a break between the setup of PCR reaction due to any disturbance, or suddenly you remember or recall something and you are out of the reaction, so you may forget that what you have done so far. So make a tick by everything, by every constituent that you add to the tube, okay? And then make sure that you have added all the 8 components into the tube. And the other thing is that you can look at the total volume, that what volume of PCR reaction you are setting up. Most oftenly in the laboratory, we are setting up a 25 microliter reaction, okay? So calculate from the concentrations of each of these ingredients that how much you need of this material so that you get a 25 microliter total volume, okay? The quantities of these materials, including target or the template or the primers or the polymerase or the nucleotides, magnesium chloride and buffer, they can be very easily calculated, and they usually go around 20 microliter. So you are left with 5 microliter, and you make up the volume to 25 microliter by adding the water. If you end up with 18 microliter, obviously you will go for addition of 7 microliter water to bring the volume to 25 microliter, because all the calculations for these ingredients, they are for 25. Okay, so once you have set up your PCR tube, you have added 8 components, there are chances that you can introduce some bubbles in it. So you can give your tubes a little spin just for 15 seconds, and all the bubbles will be removed. All your PCR ingredients will settle down to the bottom of the tube. Okay, Now I will state here one more thing, that while you are performing a PCR reaction, you are mixing a number of ingredients, and after that you need to mix them. Okay, this is highly essential. What about in PCR? Do you really need to mix them? Mixing can introduce some bubbles. It can cause some oxidations. There's no doubt about it. Okay. But remember, you are going to high temperature, 94 degrees centigrade, which is very close to the boiling point of water. And high temperature causes a lot of movements of molecules within that PCR reaction. Even you forget to mix it. That heat will mix that all for you. Okay? So just mix a little in the beginning, just a little. Make sure your polymerase has been suspended well, because it has got 50% glycerol. So it will tend to settle down in the bottom of the tube. So you will just make sure that your polymerase is well suspended. Don't worry about all the rest of the components. Give it a little mix and then spin it for a few seconds. Everything settle down, and then you can go to a PCR machine and you can set the PCR machine to 3 different cycles or 3 different uh, parts of the cycle. These parts are denaturation, annealing, and extension. All these T temperatures, they are different. First is 94. It is always 94. If you are using tag polymerase, it is always 72. If you're using PFU, it is always 68. Okay. So remember, these temperatures are constant. And the middle temperature, which is annealing temperature, it is different. Why it is different? It is different because in every time when you do PCR, you may be using different kinds of primers. And each different primer has a different melting point, and so a different annealing temperature. Okay, so this one is variable. The annealing point is variable, while the denaturation and extension temperature, they are constantly set at 94 and 72 degrees centigrade respectively. So here it is shown that this is actually one cycle. And then you are again repeating it, temperature will go high, it will go to annealing temperature, it will again go to extension temperature, the second cycle will be completed. And the machine keeps on increasing and decreasing the temperature as you feed into the machine. That this is my denaturation temperature, this is my annealing, and this is my extension, and I want to perform 40 cycles. Okay, with this information, we start the machine. Okay, by putting in the
सवाल था
जी पूछिए
सर, जो आपने बताया कि पीएफयू के लिए जब एक्सटेंशन पे जाते हैं तो टेम्परेचर 68 हो जाता है और टैग पॉलीमरेज के लिए 72, तो क्या
जी
डीनेचुरेशन में भी पीएफयू के लिए जो टेम्परेचर है उसमें कोई चेंजेस आते हैं या 94 ही रहता है?
94, because it has nothing to do with PFU, it has to do with template DNA. We remove the secondary structures. Okay, so denaturation remains the same. The extension is dependent on the kind of enzyme you are using. Okay, usually either you use 72 when you are using tag polymerase, or it is 68 if you are using PFU DNA polymerase. But you must remember, PFU incorporates nucleotides at a much slower rate than tag polymerase. If you read 1 minute of extension for tag, you will need 2 minutes for PFU. Double your time of extension if you're shifting from tag polymerase to PFU polymerase. Is it clear?
Yes, sir.
Okay. Now I move to the next slide. And this is the heart of PCR. Okay. If you understand this, I would say that you are not a technician, you are a real scientific personnel. Okay? Now, I have mentioned it several times that when we are on an interview table, either it is a viva or it is an interview table, we asked the students or the interviewer, the candidate, that how you set up your PCR reaction. So you know what they start telling? They start telling, okay, I used 1 microliter of template, I used 2 microliters of primer, I used 3 microliters of Dntp, 1 microliter of polymerase. This is the typical answer given by a technician, okay? And you must understand that how you would answer that, okay? I said that we perform a 25 microliter PCR reaction. In that PCR reaction, you do the calculations in the beginning, okay? Calculations for what? Calculation for all these ingredients. Your template should be in the range of 0.5 to 50 nanogram. Okay? You cannot calculate on the basis of microliters here. अगर आपका वो 0.5 नैनोग्राम से कम है 1 माइक्रोलीटर में, तो आपका 1 माइक्रोलीटर नहीं चलेगा. So you need to calculate that how many copies of the template you need. Now, remember my sentence as a rule. And as a rule, you must have 1,000 copies of your template in your PCR reaction if you want to have a real good amplified product. You must have 1,000 copies of the template, the gene that you want to amplify, or any other DNA fragment that you want to apply. You must be sure that there are at least 1,000 copies of that. Now, if you have cloned the gene into a vector and you have isolated that plasmid, which is recombinant plasmid, having the gene of your interest, and your template is the plasmid, you will be having millions of copies per microliter. You don't need to worry. You need to dilute that plasmid to have lesser number of copies, 1,000 number, 2,000 number in your PCR reaction. You need a dilution if you are using a plasmid. The rule of the thumb is that you isolated a plasmid, you run it on the gel, take 1 microliter of your purified plasmid, run on the gel, and if you see very clear bands of the plasmid, make a 1 to 20 or 1 to 50 dilution of that 1 microliter. And from there, use 1 microliter in the PCR reaction. Don't use 1 microliter directly from a plasmid preparation. It is too much template. Okay, so why to waste that? Make a dilution 1 to 20 or 1 to 50 and use just 1 microliter from there. You have millions of copies in your purified preparation. If you make 50 times dilution, you are still getting a lot of templates. But the problem comes that when you move from plasmid to chromosomal DNA, remember the size of the genome, the human genome, is 3.2 billion nucleotides. Out of these 3.2 billion nucleotides, only 5% of those, they code for genes, and the rest of them, they are repeated DNA sequences or regulatory DNA sequences. Okay? That 5%, it means 95% of the DNA which your taking in a genomic DNA, it is of no use in the PCR reaction. The gene will be amplified by only from some of the DNA which is there in 5% of the genome. How many genes are there in the human beings? About 30,000. And your gene is just one of them that you want to amplify. So that 5% of the DNA has 1/30th part of your gene. Okay? Now let's take it away. The total DNA in a cell, in the diploid cell, is 6 microgram in human beings. 6 picogram. Microgram is a big figure. I just said that by mistake. 6 picogram. 1 microgram has got 1,000 nanograms. 1 nanogram has got 1,000 picogram. So you can now think, what would be 1 nanogram? Very minute quantity of DNA. Okay, 1 picogram is a very small value. A single cell has got 6 picogram. And 6 picogram, if it is from the diploid genome, it will contain 2 copies of the gene of your interest. How many genomes you need to get to the 1,000 templates? 1,000 divided by 2 is equal to 500. Okay? You need 500 genomes to present 1,000 template molecules. अब आपको कुछ समझ आ रही है? 6 picogram,
Yes,
6 picogram of DNA has got 2 copies. How many picograms? They will have 1,000 copies. 6 multiplied by what? Or you divide 1,000 with 6. Okay, you get a value and multiply it with 6 then. That is the amount in picograms that you need to achieve 1,000 template copies. Okay, that's the reason you see a range. This range is towards plasmid, this is towards the genomic DNA. If you're working with genomic DNA, you need much higher concentration, okay? Which is sometimes 50 nanogram and sometimes even more than that. It depends on the genome size of that particular organism. Humans have got 46 chromosomes and cotton has got 54. Much higher amount of DNA per nucleus. Some other plants, wheat, consider wheat, it's an hexaploid. It has much more higher concentration of DNA in its single genome. So you need to calculate 1,000 copies per genome. How you can do that? Who will tell you what is the amount of DNA per nucleus? Just go to Google and ask the Google, amount of DNA per cell in cotton, amount of DNA per cell in wheat or some other organism, and you will get the answer. Okay? So all these values are known. You need to get 1,000 copies, and that is very essential for a good PCR amplification. Okay, make it a rule that start your PCR with a minimum of 1,000 copies, okay? And then the amplified product will be visible on the gel after 40 cycles, okay? Otherwise, the visibility will be very, very difficult because the number of amplified products will be very, very low. Oligonucleotide primers. What concentration? Not in microliters. Okay. What's the concentration that you will use? When you get a primer synthesized from a supplier, what you get? You get a tube having a solid pellet, which is very, very small. And then you get a sheet, a paper sheet with that, synthesis report. It's called synthesis report. And it will tell you that this tube has got 146 microgram, for example. So what you will do? What I do in my lab is, if I get 146 microgram in a tube, I add 146 microliter of water to dissolve it. High quality water, obviously. In molecular biology, whenever we talk about water, it is high quality water, which is sterile as well. If I add 146 microliter, what is the concentration of the primer per microliter? The total is 146 microgram. It is 1 microgram per microliter. Okay? That is our stock. And then what we do? If we need 25 nanogram per PCR reaction, and if we take 1 microliter, it will be 1000 nanogram in there. No? Is it clear?
Yes, sir.
Okay, so how much dilution we need to make of our stock to use 1 microliter? 1 microliter from the stock has got 1000 nanograms. We want 25 nanogram per microliter. How much dilution we will make? Divide 1000 by 25, that is equal to 40. Okay? It means we need to dilute that 1 microliter from the stock, which is having 1 microgram per microliter concentration. We need to dilute it 40 times. It means take 1 microliter and dilute it in 39 microliter of water, and that gives you 25 nanogram per microliter. And then you use 1 microliter from there into your PCR reaction. So your PCR reaction contains a total of 25 nanogram of the primer. Okay? And similarly, the Dntps. So don't mention about microliter. Well, a microliter may mean 1000 nanogram per microliter. It may mean 25 nanogram per microliter. So don't talk about microliters. Talk about total concentration of the primer in your PCR. That's 25 nanogram. And your DNA, also in nanograms. If you use plasmid, say 0.5 to 1 nanogram of plasmid. If you used genomic DNA, say 50 nanogram, 60 nanogram, or 40 nanogram of the plasmid. Don't answer it, microliters. Okay, so this is the take-home message. Now about the Dntps. Okay, I will explain this one and the rest of the things. They are very, very simple. Dntps, they come as 100 millimolar solution each.
सर एक सवाल है
जी जी पूछिए.
सर, वो जो आपने एक्सप्लेन किया था कि जो हमें प्राइमर की ट्यूब आई है 146 माइक्रोग्राम्स की, और फिर उसे हमने 146 माइक्रोलीटर्स में डिसॉल्व किया था, तो लेट्स से कि अगर हमारे पास 142 की आ गई है ट्यूब माइक्रोग्राम्स की प्राइमर की, तो फिर हम 142 माइक्रोलीटर्स वाटर में ही उसे डिसॉल्व करेंगे.
जी 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 जी. Because we need 1 microgram per microliter. So जितने माइक्रोग्राम्स में आएगी उतने माइक्रोलीटर ऑफ वाटर आप ऐड करेंगे. ठीक है? So it will give you 1 microgram per microliter. ठीक है? Okay. Now look at Dntps. DNTPs come as stocks of 100 millimolar each. It means there are 4 nucleotides, we get 4 different tubes. And each tube has got 100 millimolar of deoxy ATP. Another tube, it is 100 millimolar of deoxy TTP, similarly deoxy CTP and GTP. We have 4 tubes. We need DNTPs together. So what we do is that we take 25 microliter from each tube and pool that in one tube. So we took 25 microliter from each. What would be the total volume now in a single tube?
100.
100 microliter, okay? And what would be the concentration of each DNTP in that 100? We took 1/4 volume of each one. So divide the total concentration by 4. 100 millimolar divided by 4 is equal to 25 millimolar each. समझ में है?
Yes, sir.
Okay, because when we mixed one DNTP with the other ones, obviously we diluted that DNTP. Okay, so we combined 4. So it means that every DNTP had a 4-time dilution. It dropped down from 100 millimolar to 25. So what we do? This is our stock. The actual stock is 100 millimolar. This is our secondary stock. Then we make a tertiary stock. We have 100 microliter now of 25 millimolar each. We take 20 microliter out of it and we would make 1 to 10 dilution. We add 180 microliter of water to 10 microliter. So what will be the final concentration of 4 DNTPs of a 10-time dilution? 2.5 millimolar. From 25, we are doing 10-time dilution. It is now 2.5 millimolar, and that is our working solution. From there, we take 1 microliter. And then we put that into a 25 microliter PCR reaction. 25 times more dilution. Divide 2.5 with 25. And that will be the micromoles of the DNTPs that we are using. Then we take tech polymerase comes as 5 units per microliter, okay? We can use 1 unit in a PCR reaction, but to be on the safe side, because we don't know that how much shelf life of the polymerase is left in the tube, we take 2.5 units. It means half a microliter of the tag polymerase per 25 microliter PCR reaction is sufficient. And then magnesium chloride comes as 50 millimolar solution. We use 3 millimolar. 3 millimolar is the most frequently used because it gives you optimum binding of the tag polymerase with the template primer complex. Okay. So keep in mind, magnesium chloride, the most optimum concentration is 3 millimolar. Okay. And if you ever go to PCR reaction, from that 50 millimolar, take a volume that makes a final concentration of 3 millimolar in your 25 microliter volume. And then the buffer, 10x buffer, it means in 20 microliter, you will use 2 microliter for making the dilution from 10x to 1x, or 1 microliter per 10 microliter, or 2.5 microliter per 25 microliter. So if your buffer is 10x, you use it as 2.5 microliter per 25 to make it 1x. Okay? So remember that. These are some of the important things that you need to understand for a very good PCR reaction. Otherwise, if you set up like microliters, you will not be able to fix these concentrations. You can go much higher, you can go much lower. So remember, this is your reference slide. You can use that when you go for a PCR setup in future. Okay. Okay, so magnesium chloride, it is a cofactor. Magnesium is a cofactor, as I already mentioned. Okay, and then it binds DNA and affects primer template interaction. A higher concentration of magnesium chloride leads to non-specific binding. Okay, don't go beyond 3 millimolar. If you go beyond 3 millimolar, you will get a good amplification of your desired product. But same time, you will see other DNA fragments are also getting amplified. So it will not be a pure amplification. Okay, avoid higher concentration of magnesium chloride. Magnesium also influences the ability of Taq polymerase to interact with primer template sequence. So it enhances its interaction. But don't go beyond 3 millimolar because more magnesium leads to less stringency in binding, means non-specific. Okay, so the next is Taq DNA polymerase. You already have learned about Taq polymerase. You have already learned about PFU DNA polymerase, the DNA modifying enzymes. I don't need to elaborate that again. And the only thing is that Taq polymerase is obtained from Thermus aquaticus, okay, a hot spring organism. The extension temperature I mentioned for Taq polymerase is 72 degrees centigrade. This is the temperature where the Taq polymerase is highly active. Okay, it incorporates 1,000 nucleotides per minute. 72. If you come down to 55 degrees centigrade, the Taq polymerase does not stop incorporation of nucleotide. It keeps on incorporating the nucleotides, but at a slower rate. The rate goes down to 650 nucleotides per minute. Now you come to 37 degrees centigrade, and the rate of incorporation of nucleotide by Taq polymerase is 1.5 nucleotide per second, or 3 nucleotides per 2 seconds. It means that even at 37 degrees centigrade, it is adding nucleotides, but the rate is very slow. Now, if you come down to 22 degrees centigrade, your laboratory temperature is usually at 25. Okay? If you perform at 22 degrees centigrade, you are incorporating 1 nucleotide per 4 seconds. It means even at room temperature, the Taq polymerase has started polymerization. At room temperature, the primers, they will bind non-specifically to a lot of places on the template DNA, especially when magnesium chloride is there. The extension product or that extension product will actually act as a primer when you will start PCR reaction. You will have a much higher chances of non-specific amplification. Okay, so perform your PCR reaction on ice so that you have almost negligible chances of non-specific binding of your primer with a template and extension by the polymer.